Street plate technique is used for the isolation of the bacteria into a pure culture from a mixed population. So it is a very important method for a microbiologist. Let's begin with the culture media preparation. Take weight of agar media and mix with distilled water following the label on the media container. Sterilize the media according to the manufacturer's instructions. For sterilization, some media need to just boil at 100 degrees Celsius, some need autoclaving at 121 degrees Celsius and 15 pounds for 15 minutes. Pour the hot media in sterile petri dishes aseptically and cool to solidify the agar media. Now incubate the newly prepared culture media for checking the contamination. And choose the fresh plates for testing. Now we are going to main test for inoculation. Clean our workstation inside the biological safety cabinet. Bring a bacterial culture into it from which we want to get pure bacterial colonies. And bring also a previously prepared fresh agar culture media. In this test, we will use tryptone soya agar, which is a universal growth medium for almost all bacteria. Heat the loop until it becomes red hot, then immediately place it inside the cabinet. Wait to cool the loop at room temperature. After 5 minutes of cooling, take the loop and start streaking in zigzag style on the first quarter of the plate, starting from the bacterial smear, as shown in this video. Close the plate and burn the loop again as done previously. After burning, bring the loop inside the cabinet and cool to room temperature. Streak again on the second quarter of the plate, touching the streaking lines of the first quarter. Once it is done, start streaking on the third and fourth quarter in the same way without burning the loop. This pattern of streaking is called 4-quadrant streaking method. Finish the streaking by covering the newly inoculated TSA plate with its lid. Invert the plate and incubate at 37 degrees Celsius for 24 hours. After incubation, take out the plate from the incubator. Now we are in the last step, which is result observation. Place the newly bacterial cultured plate on the stage of a colony counter. We can see the colony density is higher in the first quarter of this plate and lower in the fourth quarter. So the colony density is decreasing from the first quarter to the fourth quarter. There are many isolated bacterial colony in the third and fourth quarter of the plate. We can get a pure bacterial culture by taking a single isolated colony from this plate and streaking onto another fresh TSA culture plate.